Hi, I'm Alex.
And I'm Brittany.
And we will be explaining the process of keratinization for anatomy and physiology of farm animals.
To start off, the skin forms two layers, the epidermis and the dermis. These two are separated by a basement membrane, and the hypodermis is a subcutaneous layer.
This is the epidermis, and this is the basement membrane. This is the dermis, and this is the hypodermis.
In further detail, the epidermis is the outermost layer of skin and is composed of stratified squamous epithelium.
It is composed of 3 to 5 layers of epithelial cells. A thin layer of skin has 3 layers, and a thick layer of skin has 5 layers.
The epidermis is also avascular and depends on a blood supply from the dermis. Keratinocytes are epithelial cells that produce keratin. Keratin is a tough, waterproof protein that gives skin its resiliency and strength. Keratin is also the main component of integumentary structures.
Keratinocytes along the basement membrane of the epidermis are well nourished from the nourishments coming from the blood supply from the dermis.
Once again, this is the epidermis, the basement membrane, and a blood supply from the dermis.
Keratinocytes also have a nucleus and can divide and produce new keratinocytes. Meanwhile, that keratinocyte has been producing keratin. Once the cell reaches the surface of the epithelium, all that remains inside the cell is keratin, which means that the organelles, including the nucleus, have been dissolved.
Once again, going over this diagram, this is the epidermis, the basement membrane, and the dermis. This top layer here is the fully keratinized epithelium, meaning it has no nucleus, no organelles, it can't divide, it's filled with keratin, and sheds to make room for more cells.
These cells here are the keratinocytes. They have a nucleus, they're fully functioning, they can divide, and as they divide, they push towards the surface.
Thank you for watching our YouTube lecture for Anatomy and Physiology of Farm Animals.
We hope this helps clarify keratinization in better detail for you.
We are now going to talk about skeletal muscle contraction. Skeletal muscle contraction depends on a nerve impulse. Neuromuscular junction is the site where the ends of nerves connect to muscle fibers.
Go over this diagram. This is the neuron. These are the synaptic vesicles over here. This is the sarcolemma, this wavy part. These are the receptors, and this whole part is the neuromuscular junction. The nerve terminal contains synaptic vesicles. The synaptic vesicles contain acetylcholine, which is a chemical neurotransmitter.
When a nerve impulse comes down a nerve cell, it causes the release of acetylcholine, which quickly binds to receptors on the sarcolemma, which is from synaptic vesicles. When acetylcholine binds to receptors, a signal is sent to the sarcoplasmic reticulum. The sarcoplasmic reticulum, or known as the SR, is the endoplasmic reticulum in muscle fibers. It also stores calcium.
When the signal reaches the SR, calcium is released from the SR into the sarcoplasm, which is the cytoplasm of the muscle fiber. The calcium binds to troponin, which is the protein present on the actin filament, which acts as a receptor for calcium. When calcium binds to troponin, it causes a shift in tropomyosin, which unblocks cross-bridge binding sites. Tropomyosin is a protein present on actin that blocks the cross-bridge binding sites, or when Ca2 is low, thereby preventing muscle contractions.
When cross-bridge binding sites are open or unblocked by tropomyosin, cross bridges are present on myosin and can bind to cross-bridge binding sites on actin, which is also known as cross-bridge formation. Cross bridges are tiny levers on myosin that bind to cross-bridge binding sites on actin. Cross bridges are cocked back and pulled forward, drawing the actin filaments over the myosin filament. This results in the shortening of sarcomeres, and muscle contraction then takes place. Muscle contraction is the combined shortening of all sarcomeres in muscle fibers. This up here is the troponin. This is the tropomyosin. This is the cross-bridge binding sites, and this is where the calcium binds.
Then the tropomyosin shifts. These are the cross-bridge binding sites, and they are exposed. These are the cross bridges, and this is the myosin.
When the nerve impulse is removed, acetylcholine is no longer released from synaptic vesicles. With no acetylcholine binding to receptors on the sarcolemma, there is no signal to the SR. The SR uptakes calcium from the sarcoplasm.
With no calcium to bind to troponin, tropomyosin shifts back and covers the cross-bridge binding sites on actin. Cross bridges on myosin are released from the cross-bridge binding sites on actin. Actin slides back over the myosin, which causes sarcomeres to lengthen, which leads to relaxation. Muscle relaxation is the combined lengthening of all sarcomeres in a muscle fiber.
Both muscle contraction and muscle relaxation require energy. Energy is supplied by adenosine triphosphate, also known as ATP. ATP is produced by the mitochondria found in the sarcoplasm of muscle fibers. ATP has 3 phosphate groups.
When one phosphate group is split off from ATP, forming ADP, also known as adenosine diphosphate, a considerable amount of energy is released. The energy powers cross-bridge binding, the sliding of actin towards the center of the sarcomeres, which powers cocking back and pulling forward of myosin cross-bridge head.
ADP is converted back to ATP by the creatine phosphate. Resulting in ATP, it can be split again by releasing energy to power the uptake of calcium by the SR, also known as muscle relaxation.